Sure. Thank you, Dr. Mayan. Uh, as he mentioned, I'm Daniel Clarke. I'm a data science analyst in Dr. Mayan's lab at the Mount Sinai Center for Bioinformatics. I'm also a member of the Common Fund Data Ecosystems Links project and also the Data Resource Center. Um, and today I'm going to be demonstrating how you can construct reusable data analysis workflows without coding using this platform we developed called the Playbook Workflow Builder. So this work was recently published in PLOS Computational Biology. So definitely give it a read if you find this talk interesting and want to learn more. Throughout the talk, I'll be covering how to use the platform uh, in real time. The Playbook Workflow Builder lets you investigate data or biological entities like genes or diseases, powered by an ecosystem of bioinformatics, web apps, and microservices. It's publicly available and free to use at playbookworkflowbuilder.cloud. And really, the easiest way to explain the platform to you is for you to see it in action. So I'll be demoing it live and showcasing its various features as we go, and hopefully all goes well. As a motivating example, we found this preprint published in February. It involves some mouse models of basal cell carcinoma. Basal cell carcinoma is the most common human cancer, and it's thought to be caused by sun exposure at a young age. It's established that basal cell carcinoma is driven by hyperactivation of this hedgehog smoothened signaling pathway. And they found in this study that the G alpha subunit and protein kinase A will negatively regulate this hedgehog signaling. So mutations in these genes causing a reduction of these uh, of this activity can actually cause the hedgehog signaling to increase, thus resulting in these types of hedgehog signaling pathway, even in the absence of this gene SMO. Um, so that's kind of what they did. And they showed that uh, in these mouse models, when they cause this reduction, they observe similar basal cell carcinoma style tumors in these mice. So let's take a look and try and reproduce or reanalyze the data that what they went over in the study. So to do that, this is the study on Geo. So let me zoom in a little bit. So in the study, they again have these various mouse models. They did single cell RNA sequencing and also bulk RNA sequencing. And we're going to be analyzing the bulk RNA sequencing data from the study. If I look at the samples, there's 21 of them. Um, this this is the smoothened kind of classic basal cell carcinoma mouse model where these con samples are controls and the BCC samples have developed basal cell carcinoma. We have the G alpha subunit sample knockout samples. And then we also have this inhibition of the protein kinase A versus the controls. So these three different groups represent uh, genomic signatures that we can analyze. So let's start analyzing this data. And if we scroll down here, we can see that they offer the gene counts already processed for us. So I'm going to download this, but note that we could also go to SRA to get the original FASTQ files. And the playbook also has a way to align FASTQ files to get this type of gene count matrix file. So when I open up this file, so when I download it, This is what we get. So we get this gene counts.txt. So if I open this with Excel, we can see this is what this data file looks like. We have the different samples corresponding to the controls and the various mouse models and the gene counts uh, here in the middle. And the way the playbook works is that it expects the first column to be the gene symbol and the remaining columns to be these gene counts. So what I'm going to do is simply remove these columns. This one as well. So now that I have a matrix that looks like this, I can export this file as a CSV. I'll put it on my desktop. And now we'll go to the playbook. So this is the playbook, as I showed before. And what we have on our hands is called the gene count matrix. So if I click on upload a gene count matrix and I say choose file, I go to my desktop. I can see this is the one I just created, the gene counts 2. I had done this before, that's why we have both. So we'll take the gene counts 2. I'll submit this. And when we do this, we're shown like what can we do with a gene count matrix. If I would click back here, we can see this gene count matrix loaded in, a preview of this data frame. And we could also share this link to someone else where they could download it and actually continue. So this kind of breadcrumb here at the top is how you navigate this interface. And we're essentially extending from this file that we begun with. And one thing we want to do is manually label these samples. This is going to be a prerequisite for doing differential expression analysis. We essentially need to say what are the samples that we're comparing. So here we can see all the samples, again, corresponding to controls and these various mouse models. So I'm going to start with SMO, the classic hedgehog smoothened mouse model. Basically, they are labeled with this A. So all the ones that are A and have SMO will be perturbation, and the ones that are A and control will be control. So let's do that. And then hopefully I've got all of them. We have one more. And yeah, so once we've done that, we can label this. So I'll call this SMO versus control and submit this. From here, we have a number of things we can do. Now that it's been annotated, you can see that we can do differential expression analysis via characteristic direction, via DESeq2, and also with lima-voom. We can also get a PCA plot from this data. So I'll do that first so we can get a sense of what we're looking at. So you can see here that these samples in blue are the control samples, and the ones that I labeled, the SMO mouses, are here. And basically, you would find that if you did this and labeled all the controls, they're all showing up here in the PCA space, corresponding to this like second principal component, actually separating the controls from the group that has something that looks like basal cell carcinoma. With that, I can actually step back to the samples and extend again from here. But this time I'm going to do differential expression analysis. So let's use lima-voom. This applies this voom normalization followed by lima, which is a pretty standard way of doing differential expression analysis with bulk RNA-seq data. Once this finishes loading, we can see that we have the statistic that we get for how differentially expressed these genes are compared to the control. And you can also see that there's a lot of significant hits. If we extend from here, you can see that we can build this like classic volcano plot, which you may be familiar with if you've done this type of analysis before, that shows on this interactive plot some of the differentially expressed genes, both upregulated and downregulated genes. All right, so the next thing we can do is we can say, okay, let's take just the upregulated genes in the signature. So from the signature, we can get upregulated genes. And now that we have a gene set, there's a variety of different tools that operate on gene sets for the purpose of enrichment analysis for the most part. If I use Enricher for enrichment analysis of this gene set, we can come here to pathways, and in the study, they also used KEGG, and we can see that basal cell carcinoma, which we know from the study is what we're looking at, shows up first. And hovering over it, you can see this gene SMO. I don't know if I can zoom in a little bit. Hopefully you can see the gene SMO here showing up, and several other genes as well. And we also see a little bit further down the hedgehog signaling pathway showing up here, both very significant enrichment results, which is what we would expect from this. So another thing we could do with this gene set to like try and figure out like how maybe we could treat this is we could apply this this tool called Link's L1000 signature search. And this is a relatively recently published tool that does, it uses the Link's L1000 data set, which has chemical perturbations and also CRISPR knockouts. So this will essentially allow us to find drugs or CRISPR knockouts which would potentially downregulate these upregulated genes in order to treat this particular condition. So if I click on down here, and we see that there's some of these preclinical, like non-FDA approved compounds with not very good names, but we can also click this approved and then only see FDA approved drugs, drug perturbations that were effective at reversing the expression of these genes. And we can see a bunch of candidates, which I won't dig too far into, but these types of drugs are certainly known to be used in cancer treatment. That's kind of the extent I'll go with this particular branch. But one thing we want to do is look at these other sets as well. So to these other comparisons. So what we would do is we would come back here and then relabel to go through the other ones. And to save us some time, I've done this already. So if I come here, we can see we have kind of the workflow we had before, but now I've annotated this B section as the G subunit. And then we also have the other C samples. So these two different samples or two different mouse models we have differential expression produced for both of them. And then finally, we have gene sets from all three of these. So what we can now do is a form of consensus. So if I hold the Shift key and I click on these three gene sets, we're able to group these three gene sets into one object. And if I come here, I can rename this and just make it shorter so it's easier to look at. But basically, it has the whole provenance kind of in the description of these terms. So let's rename them. And now with this gene matrix transpose, which has these upregulated genes from these three different comparisons, we can actually we can visualize this in various ways. So one way we can do it is with this so-called superven diagram. And superven shows that there's 704 from this group, 238, 239 from this group, and 355 from this group. And you can select these cross sections, which correspond to overlaps of genes between the three different conditions. And so we see that there's 38 genes here that are like intersecting between all of them. And this essentially, since we know that all three of these exhibited basal cell carcinoma type tumors, we can expect that these genes correspond to the pathways we're looking for. So we can actually get these genes by coming back to the gene matrix transpose and computing the intersecting gene set. So when we do this, you can see we get those 38 genes as a new gene set object, which we again can apply the same types of gene set operations we did earlier in our workflow, but now on this intersecting consensus gene set. So if I again do enricher-based enrichment analysis and come here to the pathways, you can see we have a pretty strong signal for the hedgehog signaling pathway across multiple gene set libraries, not just KEGG. And if we go into KEGG, we can see basal cell carcinoma is a little bit further down here, but still significant. And as is the hedgehog signaling pathway. Interestingly, in both basal cell carcinoma and the hedgehog signaling pathway, these two genes, PTCH1 and 2, are showing up. And one thing we can do is, if we didn't know that patch 1 and patch 2 were essential to the hedgehog signaling pathway, we could select one of these genes, say patch 1, and then we can use the gene set foundation model card to get some information about this. So looks like we have a maybe not. Okay, let's try the other one. So the LLM deep dive gene summary. I don't know. Sorry, guys, we got the demo gods. But it's here though, so we'll fix that. But essentially, what we're looking at here, patch 1, this is we know to be very important in the hedgehog signaling pathway, and you can see that it's mentioned here in this summary. This summary is actually an AI-driven overview. Essentially, what we've done is we take the top 50 most cited paper abstracts that mention the gene. We give it to GPT-4o, and we ask it to summarize and cite the papers that it actually used to build the summary. And here you can see that it talks about the inhibition of SMO and its use in uh, basal cell carcinomas. So, really we're finding this knowledge that we expected. Another thing that this site has further below is these GSFM predictions. So this is a foundation model that was trained on many gene sets from the supplemental material of literature over millions. And well, I guess almost a million. Will be over a million soon. And essentially what it is trained to do is predict the genes that we've hidden from the gene set. And we then apply this to known gene sets, and then we're recovering information. So you can see that the model is able to recover the patch1 gene in the SMO, even though in this particular case, it didn't have the gene to begin with. It recovered it. And it also predicts some other stuff that we didn't actually know. One thing that I noticed in KEGG is it recovers basal cell carcinoma, and it also mentions HPV, which isn't yet known to be, patch1 isn't yet known to be directly involved in HPV, but it might be an interesting direction to look at. So I will move on from this. Let's see, what did I want to do next? Yes. So this is, one way to analyze this data set, find this consensus, and do this type of enrichment analysis to find new genes. One other approach we could have taken is called Target Ranger. So if I come back here to the manual label of the manual labeling, and I extend from this, this is Target Ranger, and what Target Ranger is it finds it's meant to receive RNA sequencing from tumors, and it uses normal tissues from different backgrounds. In this case, you can see we can use GTEx or we can use ArchES4. And its goal is to find genes that are overexpressed in the tumor sample, but lowly expressed in most healthy human tissues or cell lines from these atlases. So if I would click this, we get, I think, okay. Try this. Sorry, here we are. And I think that's what happened here. This is a bug that was fixed, actually. Okay, so what we get are a variety of different genes that are overexpressed in our sample, but lowly expressed in the normal healthy human tissue in GTEx. Importantly, it's an important distinction because we're not actually using the controls in our sample. So this type of approach could be used even if you didn't have controls, you just had expression from a tumor. And I won't dig too deeply, but in Target Ranger, we can also further subset this set of overexpressed genes by those which are secreted or membrane proteins, potentially becoming an avenue for developing an antibody drug conjugate, which would be a pretty rapid way to come up with a way to treat the disease. So that's all I really have for this particular workflow. But I want to also show you how we're using LLMs to help you use the platform better. It's important to note that this feature is somewhat beta, but it nonetheless works for relatively simple workflows. The workflow we just built, I think it would have a harder time with. But let's go ahead and use this example. So what we're actually doing is sending this message, show me the expression of ACE2 in healthy human tissues from GTEx to an LLM, in particular GPT 4.0. And what GPT 4.0 is tasked with doing is what we have done when we saw the different possible cards I could have clicked, given the data set. In this case, it had a gene, and now it's looking, oh, what's the next card I can click? It does this via a text-based prompting, but essentially this is what it produces. So it found that we had a gene, so it triggered the gene input card and it put our gene in for us. Then, since we wanted healthy human tissue expression from GTEx, it found a card that does this. And you can see that we get a table of that expression. Um, Finally, it's saying, oh, we can visualize it. We can use a horizontal bar plot or a vertical bar plot. So I'll just click on this vertical bar plot, uh, accepting the suggestion, and then it produces this. Importantly, this is a different way to approach the leveraging of an LLM towards the towards building a workflow. Uh, every single one of these cards was made by a human and vetted. So we're actually calling these APIs, and ACE2 has this expression. This is established and in a database somewhere that's being recovered, and then we make a plot with that information. We at no point have the LLM actually write any code for us, right? It's just choosing what we run. And these workflows are somewhat self-documenting, right? They explain exactly what is being done, and they have references on like where the data came from, what tools are being used. So there's somewhat little risk in using this type of platform towards doing data analysis. I think a cool thing we can do here is ask, can you do the same thing, but for Arches 4? And you'll see that it's given the ability to actually go back and branch in our workflow. And if all goes well, we'll see that it manages to do that. So you can see it went back to the gene and it branched into this Arches 4. So this is a different resource where we can find expression across a wide variety of diseases and conditions from GEO. That's what the Arches 4 data has. And then it asks us again how we'd like to visualize it. I'll just do a vertical bar plot again. So once we have this workflow built, now I just want to show you how once you build a workflow, you can do a number of things with it. So firstly, we have this summary that's built automatically. And you can see every single step that we do becomes a sentence in this abstract. And basically, this sentence, because of how it's just stacked sentences, this text can become rather monotonous. And so one way to improve things is by clicking on the GPT augmented summary, and we rely on the LLM to kind of restructure the paragraph a little bit. And it often does pretty well with when we have many like redundant sections, it's able to group them all together into a more coherent and easier to read paragraph. We can also title these notebooks. So I'll just call this LLM generated um, GTEX expression. Once we've given it a title, we can save it. And when we save it, this will be available in our account at this Playbooks section. We can also click on share a link, and this link we can give to anyone else. And if they would run that link, they end up seeing exactly what you see. We can also share it publicly. And when we do that, it will show up in this published playbook section in the community section so that other people could reproduce what you've done. One thing I haven't really showed you is that these workflows that you build can actually work with anything. So if I would have changed this gene and submit it, the entire workflow gets re-executed, given the new changes to earlier information. And this can be done with any workflow that's built on the platform. Once you've done this, you have like a new workflow that you could save or share independently. Um, so finally, the last thing that we can do is export the workflow. And if I click on this latex bundle in Overleaf, what this will actually do is creates automatically with the workflow we have LaTeX. And for people that are not familiar with LaTeX, it's this markup language that people are using for writing papers, and it can be rendered as a PDF. What we do is that sentence that we had before that shows up at the front becomes our abstract. We get the title that we put in. We get an introduction, and this introduction is built similarly to the abstract by incorporating various elements of the introduction based on all of the steps in the workflow that we've done. We get a method section built in a similar way, describing what was actually done. We can see here that median expression of the gene across the experiments from Geo were resolved from the Arches4 website's REST API. So we get a lot more details about what was actually done. Finally, we get like exports of the various tables that appear in each step of the workflow, and also the figures getting exported here in paper form and the references. So it's not quite a full paper on this workflow, but it's a very big start to write your own. Besides a paper, there's also a poster and also a slideshow presentation. So here is what that poster looks like. And again, these are meant to be like drafts that you could then build a scientific poster after performing your analysis with this platform. Okay, so that's all I have to talk about for the paper. Um, the next section, I just want to give you a bit of a greater appreciation for the ability of the Playbook workflow to accommodate completely unique workflows. So this is the component graph, and essentially what it is showing is the different cards that you would have had at each step to get to these different data types. So the first thing we did when we uploaded our data was to put in a file, particularly a gene count matrix. So I'm going to click on gene count matrix. And then if you'll recall from the gene count matrix, we were able to actually annotate it. And let's see if I can find the annotation. So here's annotate the annotated data. So you can see that the gene count matrix that we uploaded can be annotated, and that can get us to this annotated data matrix. From that, we can do differential gene expression, and from differential gene expression, we can get gene sets, and from gene sets, we can get a slew of other types of mechanisms of analyzing those gene sets. So this here is essentially this visual graph of how these different processes are connecting. And essentially, when we are building this platform, we only code each of these individual components and they hook into this graph. So every workflow that you build is really just a walk through this graph, and adding a single element can really open new pathways to build workflows. Um, I have here just like essentially the complete current graph of the Playbook workflow builder. You can see it's a mess. There's so many different things you can do with it. And adding any single new element really just increases the amount of things you can do by orders of magnitude. Um, so with that, I will come back to the slides. And I hope I've inspired you to consider contributing to the Playbook Workflow Builder. And there's a variety of different ways you can contribute to this project, and not all of them involve coding. So the first thing you can do is, like we've just done, construct workflows with the platform and consider saving them and sharing them publicly with other users so people can understand how other people are using the platform, and also so that we who maintain the platform can understand how you use it. Um, yeah, so remember that when you want to share these playbooks, it's best to add a title. And it's also great if you click on this manual summary, and add your own description about what the workflow is. It's also of note, you can click on these arrows in the report section to collapse or show different parts of the workflow. In some cases, you have these intermediary nodes that maybe don't need to be visible in the report, so those could be collapsed. But whatever you want to show should be shown. And once you've toggled the visibility and resaved, that's the visibility that will be seen by anyone who looks at your workflow. And again, this is the save button that you'll click to save it, and the share button is highlighted with tooltip. Those published workflows, again, appear in this published playbook section. The first section on the left has some curated playbooks that our team and the playbook partnership that initially worked on this project have developed and curated. All of them have descriptions. Many of them are described in the paper, and you can launch them by clicking on the map icon, or expanding it to review the description and launching it that way. The ones on the right are all community playbooks, and this is growing. And not all of them have icons at this point, but as we add more and more stuff, we can add those as well. But yeah, it would be great if you guys build workflows and then publish them so they show up here. Another important way you could help us with the platform is by providing feedback on this text to workflow feature that I demonstrated. Um, Importantly, it's we're really just at the surface of this type of feature. And it's really important that we get some like real biological questions and ideal workflows coming from those questions. Your prompts and your feedback will be saved whenever you click on the plus arrow or minus arrow. And eventually it's going to help us make this feature even better. So definitely consider doing that if you use this feature. Another way that you can contribute is by submitting suggestions. Uh, Essentially, every step in the workflow, when you extend, there'll be these two cards. You can suggest an algorithm or data transformation method, and you can also suggest a visualization method. And if you click on these, you'll be presented with this type of dialogue on the right that lets you either specify a component that already exists in the graph that your suggestion would fall under, or a completely novel one altogether. And then you can add a description of what that component would entail. How would you go from a gene to a ranked pathway in this particular example? Maybe through some type of external resource. Finally, if it doesn't fit as a suggestion, if you experience a bug, like we did during the talk, actually, it would be great if you consider submitting a issue on our GitHub. We'll be happy to look at those and fix them as soon as possible if they're bugs, or take your enhancement suggestions and try and implement them. Finally, it's possible to actually develop components for the playbook. Uh, there are these three types of components that we've set up the playbook to accept. There's something called a prompt component, and this is a component that is going to take user interaction into account. And some examples are just like uploading a file would be a prompt, but also like taking what the user has done and incorporating it into the graph, like this GMT annotation feature that we did, are also examples of prompts. And everything in the playbook ultimately takes some input type, it applies some function to it, and it produces an output type. So here, this prompt produces this gene, and a gene is usable in this API. So whenever you're implementing some kind of API, this is called a resolver, and the resolver will resolve some different type. In this way, any single component only thinks about what comes in, what goes out, and what it's doing. And the whole graph gets built automatically from there. Um, so these components can really be built independently. Finally, we have these view components, which tell you how you would like to build some kind of plot, or how to render various descriptions of the components or the tables. There's this prototyping interface that you'll be able to use if you develop components, and a contribution guide. And we'd love to receive pull requests from you guys. With that, I'd like to acknowledge the members of our lab. In particular, we have 10 or so new summer students this year, and I imagine some of them will be working on this project. So thank you in advance, but also thank you to the Common Fund Data Center Data Resource Center. That team is part of the contribution of this. The Common Fund Data Ecosystem Playbook partnership that helped develop it originally. Some of the projects that were involved are shown here on the right. And of course, the Common Fund and the NIH for their funding of this project. Yeah, so thank you. And if you have any questions, we're happy to answer them at this time.